Now let's discuss exercise on chapter 1, which is genetic inheritance and genetic control, part B, structured question. So we are going to start discussing on question number 3. So the first question, which is in this diagram, it shows you a figure that shows protein synthesis in a eukaryotic cells. So basically this is a eukaryotic cells. Okay, so how do you know that it is a eukaryotic cell? Because in the eukaryotic cell it has organelles that has double membrane. Okay, so this here you have the nucleus. Okay, nucleus, and then here, okay, you have to imagine that this is the nuclear core. Okay, so inside the nucleus you will have your genetic materials, which is the DNA. Okay, so this is obviously A is DNA molecule. Okay, so in your DNA you have many genes that codes for the synthesis of many different types of proteins. Okay, so basically only certain genes will be expressed, okay, in a particular cells. So example here, let's say here is gene 1. Okay, so this gene 1, for example, the information within this gene 1 needs to be used, okay, to synthesize a protein. Okay, a protein. So obviously the DNA molecule is too large to synthesize the protein that the synthesis of protein that occurs in the cytoplasm of cells. So here is the cytoplasm of cells, and within the cytoplasm you have many amino acids molecules. Okay, so the information within this gene will have to be copied in a smaller form of a molecule, which is the information is copied through the process of transcription. So X is actually transcription. Okay, okay, so the process of transcription is to produce a molecule, we call it as mRNA. Okay, so because this process it occurs in a eukaryotic cell, so the initial molecule that is produced is a pre-mRNA molecule. So B is a pre-mRNA molecule. So this molecule will copy will copy the information from gene 1, okay, into the pre-mRNA molecule, and then after that it has to undergo the next process, which is process Y, which is RNA processing. Okay, RNA processing. So the process of RNA processing, it has two major stage, which is the first one is the addition of the two ends. Okay, so at the 5' ends you will have you will add a modified guanosine, okay, which is you will add 5' cap, and then at the 3' end you will add poly A tail. Okay, so one of the significance of this process is to protect, okay, the mRNA molecule once it enters into the cytoplasm, protects the mRNA molecule from being hydrolyzed by enzymes within the cytoplasm. So the next stage of RNA processing is removal of introns. Okay, so within the DNA you have many non-coding sequence, and this non-coding sequence will also be copied in the pre-mRNA molecule. So you do not want the non-coding sequence, so this non-coding sequence, or we call it as intron, needs to be removed. Okay, so after you have removed the intron, you will need to join all the coding sequence, which is the exon, together. Okay, so this will produce a shorter mRNA molecule that consists of continuous coding sequence. Okay, so that's why as you see in the diagram, after RNA processing, the mRNA molecule will be shorter. Okay, so basically C is a mature, okay, mature mRNA. Alright, so after this mature mRNA has been produced, now the mature mRNA, which is a single strand molecule, can easily exit out of the nucleus through the nuclear pore and enters into the cytoplasm. Okay, so in the cytoplasm, uh, it will find a ribosome. Okay, it goes in between the small and the large subunit of the ribosome. So basically, another function of the addition of this 5' cap and also poly A tail is to help the mRNA molecule to attach to the small ribosomal subunit of the ribosome. So after the mRNA has attached, okay, to the small subunit of ribosome, then the next thing that happens is that an initiator, okay, tRNA, that brings the first amino acid, which is methionine, okay, will bind to the start codon on the mRNA. Okay, so after that, the large subunit of ribosome will come, and then it forms uh, uh, the the translation initiation complex. Okay, and then the process of translation will start, okay, to elongate the polypeptide chain. So basically, F is the growing polypeptide chain. Okay, so now you have the A site here. Okay, so this is the A site, so this is the P site, and this is the E site. So the A site now here is vacant to read the next codon. Okay, so the tRNA that brings specific amino acids will enter to the into the A site. Okay, according to the codon that enters into the A site. So basically, E is amino acid T RNA. Okay, tRNA. So tRNA that brings specific amino acids according to the codon on mRNA at the A site. Okay, so P site is the site where the tRNA that holds the growing polypeptide chain will be, and then the E site is the exit site, okay, for the tRNA. So basically, in this diagram, you must understand, okay, the process of transcription and also translation in the process of protein synthesis that occurs in eukaryotic cells. Okay, so for the first question, it says here, state the significance of process Y. So as you know, process Y in the diagram is RNA processing. Okay, so I've mentioned earlier, okay, in my explanation, that process Y is which is RNA processing. It is significant, okay, which is the RNA processing will help to produce to convert, okay, pre-mRNA into mature mRNA. Okay, so this will produce a mature mRNA that has a continuous coding sequence. Okay, so this will synthesize the protein molecule continuously without the interruption of the introns. Okay, so and then so this give you 1 mark. Okay, so the question is worth 1 mark, but you could also give other significant of process Y, which is process Y, which is RNA processing, helps, okay, to export mRNA molecule or mature mRNA molecule to the cytoplasm. Okay, or you could also give another significance of process Y, which is RNA processing. Okay, which helps to protect the mature mRNA from hydrolytic enzyme in the cytoplasm. So that is the significance of attaching the 5' cap and also poly A tail. Okay, so this is to protect the mRNA from hydrolytic enzyme in the cytoplasm. And also, the 5' cap will also help the mRNA to attach to the ribosome. Okay, so basically, whenever you come across a question that asks for significant, okay, so it means that the question wants you to write what is the importance. Okay, okay, of that certain topic. Okay, so in this case is RNA processing. And make sure that every time when you write your answer, you do not write your answer in this form. Okay, so this is just a note. Okay, point form. Make sure that every time when you answer question in the exam or in the test, you write a full sentence. Okay. Next question, it says here, how does process X in eukaryotic cells differ from those that occurred in a prokaryotic cell? So as you know, process X in the diagram, okay, is actually transcription. So you look at the mark for this question, it worth 2 marks. Okay, so for the first mark that you could gain is you mentioned what is X. Okay, what is process X? So process X is transcription. Okay, so in the question here, the keyword here is for you to give differences between the process of transcription that occurred in eukaryotic cells and also prokaryotic cells. So what are the difference of the process transcription in eukaryotic and also prokaryotic cells? So basically for eukaryotic cells, transcription will occur in the nucleus. Okay, and as for the prokaryotic cells, it occurs in the cytoplasm. That will give you the second mark. Okay, or you could also another correct answer. Okay, another correct answer is the process of transcription that occurs in eukaryotic cells. Thus it has to undergo RNA processing. So it means that after transcription, the process will produce pre-mRNA transcript that undergoes RNA processing, that will produce mature mRNA that will be used in the process of translation later, okay, to produce the protein molecule. But as for the prokaryotic cells, it does not undergo RNA processing. Okay, so it means that after transcription, the mRNA transcript produced, okay, can be straight away used for translation process. Okay, so that is another correct answer, but still the it gives you only 1 mark. Okay, so for these 2 marks that you could gain, either you give 2 differences, okay, or you mention what is X, okay, and then you give 1 difference. So either one you can write as your answer. For this next question, it gives you the nucleotide sequence of molecule A. Okay, so this is the nucleotide sequence of molecule A. And then the question wants you to write the complementary or determine the complementary nucleotide sequence of molecule C. So if you refer back to your diagram, okay, so molecule A is DNA, molecule C is mRNA. So this type of question, where you have to determine DNA sequence of complementary DNA strands or mRNA's sequence, or the anticodon of tRNA, usually the mark worth 1 mark. Okay, only 1 mark. Although you have many bases in the strand, okay, but it only worth 1 mark. And so you have to make sure that after you have written the complementary DNA sequence, you have to make sure that you checked your answer. Okay, so if you got one nucleotide wrong, okay, for example here instead of C you write G, okay, so your answer, the whole of your sequence will be wrong, okay, and then you will get 0 mark. Okay, so make sure you check your answer. So on the DNA that start with 3 prime, so in order to synthesize mRNA, the synthesis of mRNA has to occur in the direction of 5 prime to 3 prime. So you always have to remember to add new nucleotide to the either DNA strands or mRNA strand. New nucleotide is always added at the 3 prime end. Okay, so therefore the synthesis of mRNA has to be in the direction of 5 prime to 3 prime. So the DNA that becomes the template must start with a 3 prime here. Okay, so for example here you have a DNA molecule. Okay, so the top strand has a 3 on this region. Okay, and then on this side you have the 5 prime So the complementary strand, DNA strand, will be 5 and 3. Okay, so which of these two DNA strands will become the template to synthesize the mRNA? So obviously the top strand will become the template. Okay, so so 3 has to pair with 5. Okay, and then on the DNA you have the thymine, so on the mRNA it has to pair with adenine. Okay, so on DNA you have the adenine, so on the mRNA you have the uracil. Okay, do not write thymine. So thymine is only for DNA. Okay, so you have to replace thymine with uracil for mRNA. Okay, so this is the whole sequence of the molecule C. For question B, it gives you table 1, which is codon table for mRNA, a portion of codon table for mRNA. So from this table, you have to use the information within this table to determine the amino acid sequence of molecule F. Okay, so molecule F is a polypeptide chain. So in order for you to determine the polypeptide chain of molecule F, you have to separate the mRNA sequence, okay, into codons. Okay, so this the mRNA sequence you have to separate into codons. Okay, so separate the sequence into codons. So from there it will be easier for you to determine the amino acids coded by the codons. Okay, coded by each codons. So for example, the first codon here, it reads AUG. So just refer to your table. So the first letter is A, the second letter is U, and the third letter is G. Okay, so the first amino acids is methionine. So as you know, methionine is the first amino acids, okay, in the process of protein synthesis. Okay, so it is bought by the initiator tRNA. So the second codon, it reads CUC. So CUC is leucine. The third codon is GUA. So GUA is valine. The next codon is AUU. AUU is isoleucine. And then the last codon is UGA. So UGA does not code for any amino acid because it is a stop codon. So stop codon indicates it is the termination of translation. The process of the translation is terminated to produce the complete polypeptide chain. So remember, you have to memorize, okay, at least the 3 stop codons. Question C says, using the nucleotide sequence of molecule C, so as you know, molecule C is mRNA. So from that mRNA sequence, you have to determine the anticodon of the tRNA that brings amino acid valine to the ribosome during translation. So if you refer back to your mRNA sequence, which is 5' AUG, CUC, GUA, AUU, and UGA 3'. So from that mRNA sequence, you have determined the polypeptide chain, the sequence of amino acid for the polypeptide chain, which is methionine, leucine, valine, isoleucine, and stop codon. So for the question, it wants you to determine the anticodon of tRNA that brings the amino acid valine. So here is your amino acid valine. So the codon on the mRNA is GUA. So for your answer, the anticodon for tRNA must be, because mRNA start with 3, so anticodon has to start with, because mRNA start with 5, okay, so the anticodon of tRNA must start with 3 prime. So GUA, the complementary sequence will be C A U 5 prime. Okay, so the answer will be this 3 prime CAU 5 prime. So that is the anticodon of tRNA that brings the amino acid valine. Please take note that every time when you write an anticodon sequence of tRNA, you must always write the two ends of the anticodons, okay, the directionality of the two ends. So for anticodon, because it complements with the mRNA codon, as you can see, mRNA start with 5 prime, so the anticodon has to have a 3 prime over here, okay. So after you have written the three bases of the anticodon, at the end you must write a 5 prime, okay, because over here on the mRNA you have a 3 prime. So please make sure to get that 1 mark, make sure you write the two ends of the anticodon. Anticodon are found on each tRNA molecule. Question D. Referring to figure 5, which is codon table for mRNA, the amino acid leucine is specified by 4 codons, which are CUU, CUC, CUA, and CUG. Explain the significance of these genetic code characteristics. So if you look at the codons for leucine, the first and the second bases are the same, which is CU. But for each of the codon, it differs at the third base. Okay, so the third base can be U, can be C, it can be A, or it can be G. So this is the first characteristics of genetic code characteristic, which is redundancy. Okay, redundancy of genetic code for a specific amino acid. Or you could explain in another way, which is an amino acid can be coded by more than one codon. Okay, some of the amino acid can be coded by more than one codon. Or for the same point is degeneracy of genetic code. That is for the first mark. Okay, for the second mark is to minimize error. So when the third base is changed, this will code for the same amino acid. So it only allows silent mutation to occur. So when the third base is changed, for the codon that code for leucine, it still code for leucines. Okay, so this is to minimize error, or it avoid point mutation, or it can avoid missense mutation. Okay, or all of the four codons specified for leucine. And the last question, which is question E. So in this question, it asks you on gene population. Okay, so a population is in a Hardy-Weinberg equilibrium. So 5 out of 300 individuals are left-handed. Left-handedness is a homozygous recessive trait. So for this type of question, you have to identify or take note what are the clues given in the questions. Okay, so it says here 5 out of 300 individuals are left-handed. Okay, so left-handed is a homozygous recessive trait. So for this type of question, you have to apply the two formulas, okay, which are the first formula is p squared plus 2pq plus q squared equals to 1. And another formula is p plus q equals to 1. So for the first equation here, p squared, okay, it is to find the frequency of the homozygous dominant individuals. Okay, so it is the frequency of homozygous dominant individual. For p, for the second component, which is 2pq, this is the frequency of heterozygous, okay, heterozygous individual. So if we were to okay, put an LL here. Okay, so this one is homozygous dominant plus, okay, heterozygous plus, okay, the homozygous recessive individuals. Okay, the frequency of homozygous recessive individuals. So which is AA. So imagine these are the genotype that are present in that in that population. So the whole population must equal to 100%. Okay, so because it is frequency, so it is equal to 1. All right. So this is how you understand the formula. So and then for the second equation here is to find the frequency of allele frequency. So P is the frequency for the dominant allele, Q is the frequency of the recessive allele. Okay, so this would equal to 1 or 100%. So how many of individuals, okay, how many dominant alleles, okay, are present in that population, and how many recessive allele are present in that population. So this would total up to 100%. Okay, so in the question, it gives you the information given in the question is 5 out of 300 are left-handedness, where left-handedness is homozygous recessive trait. So this means the value given in this question is for Q squared. Okay, Q squared. Q squared is for the frequency of homozygous recessive. So for question number 1, okay, for the first question, calculate the frequency of recessive allele. Alright, so that will be easy. So you know that Q squared equal to 5 out of 300. So now you just find the value of Q, which is you square root 5 over 300, and then you will get the value of Q, which is 0.1290. Okay, so usually for this type of question, you just take for 3 decimal point. Okay, and then after you have determined the value of Q, now you can proceed to the next question. So in the next question, it says how many right-handed individuals in the population are carriers for the recessive alleles. Okay, carriers. How many right-handed individuals are carriers? So right-handed individual, right-handed is the dominant trait, but they are in the homozygous. They are heterozygous. Okay, they are heterozygous because they are carriers. So basically, you have to find or use 2pq. Okay, the value of 2, you have to insert the value for p and q in the 2pq portion of the first equation. Alright, okay, so you already know the value of q. So now it is time for you to use the second equation first. Okay, so p plus q equals to 1. So you have the value of q. So now you just determine the value of p. So 1 minus value of q, which is 0.129. So you will get the value of p, which is 0.871. Okay, so this is for 1 mark. Okay, 1 mark. And then from there, you can determine the frequency of the heterozygous. Okay, because you want individuals that has the dominant trait, but they are carriers. So obviously you want the heterozygous. Okay, you want the heterozygous individual. So you just insert all the values. Okay, so 2pq will equal to 2 times 0.871 times the value of q, which is 0.129. So this would equals to 0.224. So this gains you the second mark. Alright, so the third mark. Okay, the question wants you to calculate, okay, to give the number, how many individuals, okay, not the frequency, not the percentage, but the question wants how many individuals. So from the frequency that you got, okay, for the heterozygous, so you just times with the total population, which is 300. So 0.221 times 300, so you will get 67 people. are heterozygous. They are carrier for the dominant trait, which is right-handed. So this ends the discussion for the first exercise on chapter 1, okay, which is genetic inheritance and genetic control. So I hope you understand all of what I've discussed, okay, from the MCQ question up to question 3, okay, for structured question, all the question that I've discussed. So if there's still something that you don't understand, so you can ask your friend, you could discuss together to find the solution to your problem. Or if you have discussed together and still you cannot find the answer, okay, or you are still unsure or uncertain, you can text or WhatsApp me, okay, to ask anything that you are not sure of on this topic.